Hi guys, this is Dr. Buhler again, and we're going to talk today about the pharmacology for treatments for COVID-19 disease caused by the SARS-CoV-2 virus. It's February 2021, so we have a few drugs out there. We don't have a lot of them. Only one of the drugs that we have out there actually is fully FDA approved for use in COVID. There are several that have emergency use authorizations but aren't completely approved yet. To understand this lecture, it'll be really helpful for you to have some background in the viral life cycle of SARS-CoV-2 and some idea about disease progression and basic disease properties. It'll also be really helpful for you to have some knowledge about different types of vaccines. And so if you're not really familiar with the multiple types of vaccines, I've got a YouTube video posted that'll talk about those. So without going through all of the life cycle of replication of the SARS-CoV-2 virus, I'm going to briefly summarize it, putting the spotlight on drug targets. So the first thing that's going to happen is the docking and fusion phase. The SARS-CoV-2 virus has several external structural and binding proteins. Those include the envelope, the membrane, the spike protein, and then internally the nucleocapsid protein that winds around and protects the viral RNA. So SARS-CoV-2 is an RNA virus and it's not reverse transcriptase. So it's going to come with RNA and then it's going to use RNA the whole time. So you're going to start with the virus here in the docking phase. Docking is also called binding. It's also called attachment. So the virus has to attach somehow to the host cell. So it has this S protein, also called the spike protein, and that's going to attach to receptors on the outside of the host cell called ACE2 receptors. Because that's the way that the virus identifies the host cell, it's not going to bind on any cells that don't have that ACE2 receptor. So the virus is going to be fairly selective about what type of cells it can infect. After it's bound to that ACE2 receptor, there's another enzyme protein that's sitting out here called the TMPRSS2, and it's a protease. It's a host enzyme. It's going to come in here and it's going to cleave off part of those S proteins. That's going to allow this virus to get pulled closer to the cell membrane, which is called fusion. It's then endocytosed into the cell, which requires endocytosis structural proteins. And then it releases the contents, which is the viral RNA. That viral RNA now that's in the host cell is in the form of mRNA. So the host cell can automatically start using that viral mRNA to use the host ribosomes to form viral proteins. Those viral proteins are in the form of polypeptides, so they're long strings of amino acid, of peptide, that then have to be cut apart into the smaller functional proteins. And those are cut apart by viral proteases. All of those structural proteins then go through our own cell pathways of modification and transport and are taken eventually to the cell membrane where they're going to be released. So that's part of the pathway for production of new viral proteins. We've also got to replicate the RNA that the virus came in. So we're going from the original RNA form that entered into the cell, and we're going to have to reproduce that to new RNA that's going to be packaged into the new viruses. So our bodies don't ever need to make RNA into RNA. We either go from DNA to DNA or from DNA to mRNA. So we don't have nucleic acid polymerases that are going to do this job. So one of the genes that the virus coded for that came in was for a viral RNA to RNA polymerase. You can also call that an RNA-dependent RNA polymerase. Through that endoplasmic reticulum protein processing pathway that I mentioned, all of those viral proteins, the structural proteins, the nucleocapsid, and the new RNA are all going to end up being assembled and then exocytosed, which we also call budding, ending in the release of new viruses, also called virions. So when we look at each of these steps, these are each going to be a place where we have a theoretical drug target, that if we can interfere at those steps, we're going to have an antiviral. So the drug list for this lecture is going to include some vaccines, some antibody therapies, and those are antibodies that bind to that S protein on the virus. There's going to be some of those viral RNA to RNA polymerase inhibitors, JAK inhibitors, which help inhibit the endocytosis of the virus, inhibitors of that TMPRSS2 protease that was on the surface of our cells, and then also in the pipeline, some inhibitors of that viral protease. And while not technically antiviral, we want to remember that anti-inflammatories can also be used to decrease both pulmonary and systemic inflammation. And for this lecture, you only need to know the highlighted drugs here. You're absolutely going to want to bookmark this link. It's a frequently updated database of all of the therapeutics for COVID-19 and what phase they're in. This is what you're going to see when you pull up that database. You can look at either antiviral drug candidates, vaccine candidates, any agents that have emergency use authorizations. You can see the name of the agent, the company that's creating it, what phase it's in, say if it has U.S. emergency use authorization, if it's in phase 3, what the drug target is, or in terms of vaccines, what form of vaccine it is. So as you go through your rotations and as you go through the next years, you're going to want to be constantly up to date on what's out there and what's in the pipeline and about to be out there. This is really the perfect way to keep up to date on that. As I mentioned, there are multiple different types of vaccines, and hopefully you're already acquainted with these. We've got a couple types of whole vaccine, protein vaccines, vector-based vaccines, which in the case of COVID right now are adenovirus vectors, and nucleic acid vaccines. If you've already covered these at some point, you can just take a look at this slide for a refresher. If you've not already covered this material at some point, you should find a resource somewhere. You can always take a look at my YouTube video on vaccine types. So currently, February 2021, there are 10 available vaccines that are being in current use worldwide. These vaccines do have brand names, but unlike most drugs, we really often refer to them by the name of the company or the organization that's creating it. And I think that's because we've been following their development in real time. So we've known that Pfizer or we've known that AstraZeneca was creating a vaccine long before they actually had one ready to name. So we've already kind of been in the habit of saying we have a Moderna vaccine or we have a Pfizer vaccine, even though those are the names of the company and not actually the name of the vaccine. You can see that we either have released vaccines or vaccines that are in late stages of development for every single one of the vaccine subtypes. The vaccines that you're going to need to know are the ones that are highlighted here in red, and they're highlighted not because they're necessarily any better of a vaccine, but only because these are the ones that are going to be available in the United States. So we have live attenuated vaccines that are in development, and we have 3 vaccines that are already out there that are inactivated whole vaccines. We've got Sinovac, Sinopharm, and Covaxin. So those are the major vaccines that are being used in China and in India. A little name recognition tip is that Sino is a prefix that means China or Chinese. So any of these vaccines that have Sino in them, Sinovac, Sinopharm, and CanSino, you already know that those were developed in China. We have a couple of S protein subunit vaccinations. Novavax is one that is in fairly late stage development in the US. It's not out yet. And then Epivax Corona is Russian developed and it is out there. The vaccines that you're going to see right now have been the mRNA vaccines, and those are the Moderna and the Pfizer vaccines. Those consist of the RNA for the spike protein of the virus. And I'd recommend you watch this very short video refreshing you on mRNA vaccines. These are the 2 vaccines that at this point in time have emergency use authorization, but they still aren't FDA approved yet. The next 2 I'm going to talk about are already in use in the UK and Europe and have been submitted to the FDA in the US for emergency use authorization, but haven't gone through that whole process yet. These are the AstraZeneca Oxford and the Johnson & Johnson vaccines. And what's really important to distinguish these from the Moderna and the Pfizer is that these are adenovirus vectors. The major vaccine from Russia, that first vaccine that came out, the Sputnik 5, is also an adenovirus vector. And then the final vaccine type, DNA viruses, we do have one that's in fairly late stage development from a US company called Inovio. And so assuming that pans out, we'll be getting that too as well. Just like I showed you, that updated database on COVID drugs also talked about vaccines, and there's an additional link for another vaccine development tracker. This is too old even for me to remember, But the first satellite that was ever created was a Russian satellite called Sputnik. So when they came out with the Sputnik 5 vaccine, they named it that to kind of remind the world that they'd had the first satellite as well. And the way that I remember the Sputnik is both the Russian vaccine and an adenovirus vaccine is that Sputnik satellite looks a little bit like the adenovirus vector. So we've already talked about vaccines, and in vaccines, you're giving your body an antigen so that it can create antibodies to, in this case, the S protein of SARS-CoV-2. The antibody therapies rely on creating those antibodies outside of your body and then giving them to you. You can either synthesize or grow those antibodies, or you can use plasma from patients who have had COVID-19 and have already created their own antibodies. The mechanism here is really simple. You're taking an antibody that'll bind to that S protein. You're giving that antibody in amounts that it's going to let it come and bind to the S proteins. And I'm going to have it bind right here into this S protein here. And not only is that antibody going to tag that virus for immune attack, but it's also going to physically come in here and block the binding of the S protein to the ACE2. So if that binding is blocked, then it's not possible for the virus to enter the cell and you won't get infection. So these are binding the S protein. It can't attach to the ACE2 receptor. That blocks that docking or attachment step. We also call that the binding step. And then it can't enter the cell. So we have 2 neutralizing antibodies out there that have emergency use authorization, bamlanivimab, and one created by Regeneron, which is the company that created it, casirivimab, and endamivimab. If you look at those names, remember what I've been telling you. Any drug that ends in mab means it's a monoclonal antibody. And monoclonal antibodies cannot be given orally because they'll be broken down in the stomach. Monoclonal antibodies also have to be grown and created in a cell culture. And in the case of these 2 antibodies, they're being grown in Chinese hamster ovary cells that contain that S protein gene. So these are grown in non-human cells. They're given single dose IV, but because they're developed by injecting the antigen into animals, letting the animal create multiple different forms of antibodies, and then purifying out and selecting the antibodies that are most effective, it means that there are several steps in development that are going to occur every time they want to change those antibodies. So as new viral variants emerge, they would have to redevelop new antibodies for these. So that can take some time. The convalescent plasma has been granted emergency use authorization, but in a sense, it's not really a drug the way we look at it, because we're just taking plasma from a human being who has had COVID-19. We're getting everything that their body has done as an immune response to that infection. So they've not made just one antibody, they've made multiple different antibodies toward SARS-CoV-2. They have activated T cells that are also going to recognize it. So you're going to have multiple different components that are going to act against SARS-CoV-2. On one hand, that can be good because you have lots of different types of ammunition, but on the other hand, every single person's immune response is different. So convalescent plasma from each different patient donor is going to be different. The next drug class we have are RNA-dependent RNA polymerase inhibitors. And we already talked about how SARS-CoV-2 is an RNA virus that comes with RNA and has its own polymerase that's going to then synthesize new RNA to be packaged into the new virions. We also talked about how people don't have RNA to RNA pols, so that's a good selective toxicity. And the drugs that we have for RNA pols in different viral types are all analogs of either the nucleotide or nucleoside. And you're going to see similar drugs that are also in use in HIV. Just to remind you, when you're replicating your RNA, you're incorporating nucleosides as you go. A nucleoside analog drug is going to come in here and it's going to be wrong, and then it's not going to let that RNA chain continue. The drugs that we have in this class for SARS-CoV-2 right now are prodrugs. They come as nucleotides, and then they have to be converted into your body into nucleosides. At that point, they're going to be, as I said, inserted into the RNA chain, and that's going to block the RNA replication. Remdesivir or Veklury is the only one right now that's in common use, and in fact it's the only drug that has actually been granted full FDA approval for use in COVID-19. It looks like adenosine, and this was actually initially developed to treat Ebola, which also has an RNA-dependent RNA pol. It didn't work as well for Ebola, so they stopped working on it, and then they pulled it back out of the freezer and took a look at it for COVID-19, and it seemed to work reasonably well. This has IV dosing only, which makes it a little bit harder to give. There's one in the pipeline right now called favipiravir. It's also an adenosine analog. It's currently in trials, and it is also repurposed and is used in Japan as an influenza drug. It has a little bit of benefit in that it can be given PO. We don't know if it's going to be approved, but it is one that's in that pipeline. Another drug class that we have are inhibitors of the human TMPRSS2. And if you remember, the virus first bound the S protein to the ACE2 receptor, but then it had to use that human protease, TMPRSS2, in order to cleave that S protein to pull it in so that you got membrane fusion. A really important point is that the TMPRSS2 is human. That is on the outside of the host cell. It's not a viral protease. We have a drug that's already out there that has been already approved for pancreatitis and reflux disease, and it's called Camostat. It's in phase 4 trials right now, and if it looks good, they're going to ask for emergency use approval from the FDA. So I want you to have heard about this because you may be seeing it soon. The mechanism was to inhibit that TMPRSS2, so we didn't have cleavage of that viral S protein that prevented the fusion, and so it couldn't enter and it couldn't replicate. A benefit of this is also that it can be given PO. The next drug target is really interesting, and it's a JAK inhibitor. JAK stands for Janus kinase, and kinases are those critical enzymes that can phosphorylate proteins, and in doing so, modulate almost every cellular process. So the Janus kinase, or JAK, is a very specific kinase, and the reason that we're looking at it here is it's potentially going to do two completely separate things in COVID infection. So I'm going to draw a little line down here, and first we're going to look at this mechanism. We know for sure that JAK, as an enzyme, is necessary to mediate cellular responses to cytokines. So cytokines can be highly inflammatory, and some of the problems that we see in COVID-19 is that hyperinflammation in the lungs, and even sometimes hyperinflammation systemically when you see that cytokine storm. So having a drug that's going to reduce inflammation, particularly by reducing cytokine-mediated inflammation, is going to be really helpful in terms of being anti-inflammatory in COVID. We do use steroids like that, like dexamethasone, so this isn't the only drug that we have that's anti-inflammatory, but it is focused on cytokines. And the reason that we have that drug already is that because we know that it's necessary for inflammatory and autoimmune responses, it was previously developed to treat rheumatoid arthritis. So that's why we already have it out there. And the drug name is baricitinib or Olumiant, and that's given PO. So we know about the anti-inflammatory part. But the reason that this particular drug is so interesting is that it also has an effect on viral replication. So here where it says AAKI and GAK, we're not going to go into that particular enzyme process, but that has a lot to do and is modified by JAK. So when you have the entry stage here and you've had your S protein bind and you've had your proteolytic cleavage, now you have to actually endocytose. And I'm drawing these little lines to show the machinery that cells use to endocytose. And some of you from cell biology might remember clathrin-coated vesicles. Like I said, I'm not going to have you guys focus on the detailed cell biology steps here, but baricitinib and other JAK inhibitors are going to interfere with this endocytosis process. So even though the S protein might bind, and even though you might get proteolysis, if you can't endocytose, then you're not going to be able to pull the virus in. So the mechanism of this is twofold. It's a JAK inhibitor, which means it's inhibiting the endocytosis of ACE2 bound viruses. and it's going to block the inflammatory response to cytokines. So that's what's really exciting about this drug, is that it can have these two different mechanisms. It's been granted emergency use approval in the US. How effective it's going to be in the long run, we still don't know. Some other drug targets that we don't have drugs quite as far along in the development process, but that you are definitely going to be seeing eventually, are going to be decoy drugs, also called soluble ACE2, which basically consist of a small drug that looks like the binding site for ACE2. So that drug here is trying to mimic the shape of that ACE binding site. The idea is that that drug that mimics the binding site is going to bind to the S proteins, and then once the S proteins are bound, they're not going to be able to bind to the real ACE2. So looking like the binding site of ACE2 binds the S receptors. Now they can't bind to the real ACE2 receptors, so they can't perform that docking attachment step. Another similar idea in terms of blocking that initial binding step is to create a drug that binds and blocks the ACE2 receptor. So if that ACE2 receptor itself is then blocked, now the S protein can't bind. So those are the host ACE2 receptor blockers. And remember, those are receptors that are on the host. So we're blocking the host receptor, whereas with the soluble ACE2, we're blocking the S protein, which is the viral protein. One question about these host ACE2 receptor blockers that I don't have an answer for you yet is what's going to happen when we block our ACE2 receptors. I mean, they're there for a reason. They're involved in multiple important physiological functions. The good thing about these antiviral therapies is predictably we're only going to need to use them for a very short time. We're not going to be using them over months or over someone's lifespan. So if you can give it for a week and either keep from getting an infection or fight off an infection and then stop taking the drug, we may then not need to worry too much about the physiological effects on the ACE2 pathway. But again, we don't really know that yet. And the final drug target that you want to know is the viral protease inhibitor. And remember, you have the viral RNA. It's made into polypeptides by the host ribosome. So now you have these viral polypeptides. Those have to be cleaved by a viral protease in order to fold up and make functional viral proteins. Those functional viral proteins include the RNA pol, and so you have to have those in order to have replication. So these drugs would be inhibitors that inhibit the viral protease inside the cell. And I'm trying to really hit home that this drug is not inhibiting the human protease that's on the outside of the cell. These are completely different mechanisms. But if you inhibit that viral protease inside the cell and you stop the clipping of the viral polypeptides, then you won't have active proteins and the virus can't replicate. We have drugs that do this for HIV, so they're starting with some of these HIV protease inhibitors and looking at whether they have any activity or whether they can modify or alter those in order to be more COVID selective. So you can quiz yourself on some of these SARS-CoV-2 drug targets. And while you're only required to know the specific highlighted drugs that are already in use that we talked about, you also do need to know, understand, and be able to explain the additional drug targets. Because later this summer you're going to be in clinic, next year you're going to be in clinic, and as you go through your career, you're going to be seeing these drugs coming out in these different drug classes. So even though it might be a drug with a different name, when they come out and say, hey, there's this drug that's a SARS-CoV-2 protease inhibitor, you're going to know what that drug does. And you're going to know what that means. And if you can do that as a student, you're really going to impress your preceptors. All right, thank you. And we're going to talk about influenza agents in our next lecture.